Hey everyone, going to be doing our HIV Cure research scientist Q&A and a lab tour of Gladstone Institute's lab. For those of you who have never talked to an HIV Cure research scientist, which is most people, we're going to meet a few of them actually, and then we're going to go into a lab and they're going to show us, I believe, one or two experiments that they're actively working on that are related to HIV Cure. And then throughout, I'll be fielding your questions and hopefully answer some good, juicy questions. So I'm just going to wait right now for my first guest to join. Thanks, Seabest, for the looking sharp comment. Oh, we got to hear Hope Collaboratory is here. For those of you who watch my community member, so let's see if they can come on. Welcome, everyone. Welcome to the HIV Cure live stream.
Hi,
Hey,
Raif. Hi.
you want to introduce yourself?
Yes, for sure. I'm Patricia de Fischereux, and I am the Community Engagement Coordinator for the Hope Collaboratory.
Love the eyeshadow.
Yes, I never put eye makeup. So.
Hi, I'm Alyssa Andrew, and I'm a postdoctoral researcher here at the Gladstone working on HIV cure research.
Hi, I'm Sydney. I'm the project coordinator with the Hope Collaboratory.
Yeah.
Woo.
Yeah. We're so grateful, Raif, that we can meet with you today. And I want to start by saying that we want to acknowledge and really honor the lives of all our beloved ones that we have lost or, passed away because of HIV, and those who continue to live with the virus and face, the challenges that comes along with living with the virus, as well as we have made a lot of progress, but there's also a lot of stigma that continues to pervade. And we're all here to bring a little bit of hope, maybe, and what is happening in these days with HIV cure research. And I have some really incredible young scientists here who are going to take us on a journey in the lab, and then we'll talk with Melanie. And yeah, and kudos to you, Raif, for all that you do, your advocacy, the education that you provide your viewers. You're amazing, and we love you.
Well, thank you for that.
Okay, shall we get started?
Yeah, we
Yeah.
get started. So, hope might sound a little complicated. It's HIV obstruction by program epigenetics. Don't be afraid for the complicated terms, but basically, it's a new approach to developing a cure for HIV. And Alistair here and another couple of our young scientists are going to, give us some insight into what they're doing. And we're at the Gladstone Institute. Shall we go into the lab?
Yeah, let's go.
So for those
All
of you
right.
who are just joining, just want to let you know that we are going into a lab, a real live experiment science lab, where they're working on HIV cure research, and we're meeting some HIV cure research scientists, and they'll be answering some of our questions as well.
Okay, thanks, Raif. Let's go to the lab.
Awesome, let's go.
Woo-hoo.
All right. Okay, so the first thing we're going to do is put on our lab coat.
Ooh,
So,
fashion.
let's grab mine here. Thank you. Is it yeah. Okay. All right.
Are they all the same color? Do you get color
We
options?
don't get color options. Some people have like tie-dyed and decorated theirs.
Asking the
There
really
is one
hard-hitting
lab that's
questions.
like Barbie themed, so theirs is pink.
um,
But here you're really going into it.
is this mandatory or either way? What is it for overall?
Yeah,
Alistair,
yeah.
tell you.
So, so it is mandatory. So to work in a research lab, the minimum requirements are a lab coat and some gloves, to help protect us, the scientists, and just the general community from any of the things that we're working on, and just to be safe. And so the minimum is this, but then in different areas of the lab, depending on the research that's being conducted, there might be additional clothing options and different gear that you put on. Generally speaking, though, we refer to what we wear, lab coats, gloves, we refer to that as personal protective equipment, or PPE.
Mm.
So this is our PPE for today. I'm gonna grab some gloves.
I'm sure we're all familiar with PPE after the pandemic. So Magpie, I hope that answers your question. Eb G says we love Black women in STEM.
Woo! Okay. All right. So I guess before we, or while we're heading to where we're gonna go, I'll just kind of scan the area, show you a little bit of what the lab looks like. I can talk to you as we go
Yeah.
about myself, who I am, how I got
Awesome.
into HIV research.
Someone asked
Um,
if you wear goggles too.
Sometimes we do. So it depends on what we're working on, especially if there's a splash hazard. So if we're
Mm.
working with chemicals and things that can splash around, then you definitely want to wear eye gear. So right now we're in the general facility. You can see behind me a lot of our giant freezers. That's where we store samples, reagents. These freezers and fridges get really, really cold. And so it makes sure that we can preserve the integrity of all of the things that we need as we do our research. Um, and oh yeah, I said I would talk a bit about, oh, let's go this way. I said I would talk a bit about myself.
Yeah.
Walk right by the BSL too.
What brought you into
Um,
this field?
um, so I recently finished my PhD in infectious diseases. So I completed that 2.5 years ago. And I wanted to do research that would impact minority communities, especially
Mm.
Black communities, anything related to infectious diseases that I thought could help advance cure and treatment options in the future for people disproportionately affected. So got into HIV. There we go. And now here as a postdoc, I study the immune system and specifically how the immune system recognizes HIV and responds to HIV, with the goal, hopefully, of once we do have a cure, we have a really comprehensive understanding what the immune system needs to be able to sustain that cure. So,
Mm. Is there like one cool factoid about the immune system you've learned that you could share?
hmm. Yeah. So one thing that's interesting about the immune system, it's very effective at recognizing things that aren't supposed to be in our bodies, but it does get exhausted. And so if it does recognize a pathogen, let's say HIV, it does what it can to eliminate that pathogen. But if that pathogen persists, which is the case for HIV, the immune system gets exhausted.
Mm.
And so our immune cells become a little bit less effective at destroying their pathogen HIV. And so there are certain treatments, certain approaches that us as scientists and in the medical community can employ to help give our immune system that boost to maintain its effectiveness, to kind of keep it from getting too exhausted. So I
Fascinating.
thought.
Yeah. Girl knows I'm tired.
so, so one portion of the lab that we're gonna head into now is
Mhm.
known as a
Ooh,
BSL2
what's
lab.
BSL?
So BSL stands for biosafety level. Um, and so what that is that we have certain distinctions and certain sort of areas where we work on specific pathogens and specific questions. For BSL2 though, so you can work on a variety of different pathogens, not just HIV. But what's really special about the BSL2 is that you have different sort of um, um, mechanisms in place to help protect both the researcher, our colleagues. So we use specialized equipment, which you'll see in a second, such as a biosafety cabinet. So we do have to use sort of additional barriers. In addition to our PPE, we then have other sort of facility controls and safety mechanisms that we have to use just to be respectful and safe, not to be fearful of the work that we do.
And also, it protects your experiments in the hood as well.
Absolutely. So it keeps our it keeps our experiments contained, keeps
Mhm.
them clean, sort of separate from any other unwanted, like dust bunnies, for example.
Mm-hmm.
So we get to have our experiments done in a really controlled way so that we have really clear answers to our experimental questions.
Okay, quick question. Um, is there a BSL-1? Is that the lowest? And what's the highest level?
Yeah, BSL-1 is the lowest. So BSL-1 is everything else that we just walked through. That's BSL-1. You can think
Okay.
of the bench where you see a scientist holding a pipette. That's BSL-1. So that is the lowest level, and then it goes all the way to BSL-4. BSL-4 is the super high containment. It's actually very rare. There are only so many, like a very few BSL-4 facilities in the world. But that's where you would do the really sort of research on like Ebola or anything that is just really a serious pathogen that you have to be extra careful with. So we don't have a BSL-4 here, but we do have a BSL-3. And each BSL level has a different set of PPE, a different set of protocols, different precautions that you would follow through to make sure that you're safe and to conduct the experiment. Correct.
Okay, thank
All right,
you
so
for
let's
that.
head in. Hello. So this is our BSL-2 facility. So as you can see, we've got here some gigantic machinery here. This is known as a centrifuge. It helps us to manipulate our samples in the ways that we need. We have the biosafety cabinet, which I mentioned before. So that's right behind me, right here. And as you can see, you would do your experiments sort of in this contained area. So if there's a spill, if there's anything, it's all sort of contained within one place. We have some incubators towards the back so we can do experiments where we can control the temperatures that our experiments are conducted at. And sometimes the temperature is really important for asking a question about the virus. And yeah, so I'm going to hand it over to my colleague, Julia. She's going to talk to you about what she does in the lab, and you'll see her in the BSL-2.
Hi, Julia.
Hi, everyone. Hi, Raif. Good to see you.
Good to see you too.
So,
What you got
so
going on in here? What's cooking?
yeah, so I want to bring you to the BSL-2 today and show you some T cells, actually. So, as Elsa mentioned already, we are here in the BSL-2 laboratory, and whenever we work with HIV, we also work with T cells. So, T cells are a very unique kind of cell that we have in our body. It's circulating in the blood, and it's actually the major target cell for HIV in the body. So, whenever we're working with HIV, we also are working with T cells, kind of. And in the lab, we keep our T cells in the incubator. So these are right here at 37 degrees because we want them to be happy and cozy, warm. And we can look right into one of those incubators.
Ooh.
So we keep our T cells in these kind of tiny, sweet little flasks.
Ooh.
They have a lid on top with a filter inside, so they are kind of protected from the outside world. And to work with them, we're going to bring them right into one of those hoods here.
Do they naturally look yellow, or is that some kind of fluid that's yellow?
Yeah, so this is the cells are in like a suspension of media containing nutrients like glucose and also amino acids. So all the essential stuff the cells need to be happy, as we do as well. And when the cell is kind of growing and multiplying, the cell will kind of consume those things, and then the color of this medium will change actually from a more orange kind of thing that
I have
actually
one question from the viewers from Fox asking, do you get the T cells from donated blood, or where do they come from?
Yeah, so most of the times we're actually getting T cells from blood that got donated or from, for example, clinical trials. And then we take the blood here, actually to the same kind of setting. And then we're using special techniques in the lab to isolate only the T cells. Because in the blood, you can imagine there's like tons of other things inside. But if we're interested only in the T cells, we've got to isolate only the T cells.
Fascinating. Thank you.
Okay, so if we're working in those biosafety cabinets, what we're actually having is like a very sterile and contained environment. So now it's safe to open the flask with our T cells in here. The reason why we're doing this inside is we want to protect our cells that we're working it with from the environment. Because the environment and also our skin, our hair, everything is fully covered with other microbacteria and microorganisms and dust and everything. And we really want to protect our T cells from that. And this is why we're working inside these biosafety cabinets. Yeah. And then everything we keep in here is sterile. We spray everything with ethanol. So we have those bottles here everywhere. We spray our hands when we start working.
Is there, someone's asking, Captain Williams is asking, is there an expiration date on the T cells you guys store?
So, for example, when you're isolating T cells directly from the blood of a person, they usually can survive outside the body for like 1 to 2 months if we treat them correctly. So that's possible. Yeah.
Okay.
Are there any other questions? People,
I
come on,
think that's
ask
it for now.
questions. That's what we're
Yeah,
here
don't be
for.
shy.
If not, I'm going to continue showing you the hood and what kind of work we're doing in here.
Yes,
So
please.
let me sit down here. I'm going to spray again my hands with ethanol, so my T cells will be fine. And then I go here inside the biosafety cabinet with my hands, and I make sure that really everything from my skin is covered. So really none of these dusty things that are living on my skin is actually falling into my T cells. And then what we can do is we can open the flask. Let me take one of those pipettes here. And this is really something we all do every day. So this is really our bread and butter. We're doing this really almost every day. So what we can do is we can take one of those pipettes and we can take a couple of milliliters of this T cell suspension, resuspend them a little bit. And what I'm going to do is I will put them into one of those tubes.
This is giving Resident Evil vibes.
You say you're Resident Evil.
Okay,
And
so
then after we're gonna
I'm sorry
see
if I
some of the
missed it.
results, right? Yes. With these cells.
Yeah, in the next station, actually, with the next scientist, you're going to see some of those cells under a microscope. And
Yeah, can you tell us the objective of what you're doing here?
yeah, so right now is what we call splitting the cells, because we need to take care of the cells, right? They breathe and they consume the sugar and other metabolites that are in the medium. So we have to take care of them and give them fresh medium as well. So the way I'm doing this is I transfer part of this into those tubes. And in these tubes, we can actually spin them down, meaning we will collect the cells at the bottom of the tube, and then we can remove all the liquid above it. And then I can replace this old medium with fresh medium that I have here in this bottle.
Okay.
So this is how the medium looks like if it's fresh and new. And then this yellow color comes when the cells have actually eaten all the sugar inside.
So right now you're basically maintaining the cells and keeping them
Yeah.
alive. Okay,
Keeping them happy. Yeah.
great.
And
That's awesome.
we need all that, Raif, because this is kind of one of the building blocks of the research on HIV cure. We need to have these cells so they can do the experiments coming in with viruses that have been made or can be used in the lab for the specific experiments. So the folks here they work at all hours, endlessly, super dedicated to the research. You can see them, when they have these long experiments with the cells, they feed here overnight. They have to wait sometimes several days to get their results. really thankful for this young people, young generation,
Mhm.
and they're working on this new approach for the cure. So, you want to take us? We go to see Fran and we see some of the cells under the microscope.
Yeah.
Let's go.
Thank you so much for showing us that. How exciting and interesting. For those of you just joining, we are taking a tour through an HIV cure research lab, answering some questions. They're explaining what they're doing, and now we're going to go into one of their experiments, which is fascinating. We just saw how they store and maintain T cells, and then they're going to use these T cells to do experiments. Oh, hey, how's it going?
Hello, Raif. How's it going?
Mhm. You want to introduce yourself for the
Sure,
folks here?
sure. Hi, everyone. My name is Francisco. I'm a postdoc at Dell Lab, and I'm also working in HIV Cure. Yeah,
Awesome.
I don't know.
I think what everyone will notice too is how young all three of you are. We have such young people working on this research,
Thank you.
which is great. I mean, when you think of scientists, you think of like, an old white-haired dude, right? So,
Well, it's some more than others.
I'm sure. Yeah. Okay. So what do we have here? What are you working on?
So my work is gene therapy approaches, novel gene therapy approaches for HIV cure, and we are working in developing a new method that is DNA break free, so it does not damage the DNA. And what I wanted to show you today is one of our assays in which we figure out whether our therapies are either working or not in vitro. So basically with the cells that we are growing. So we look here at the screen, you will see a bunch of black dots. Each of these black dots is actually a T cell. And you will see then, I hope you can appreciate
Can
that
you
some of these
angle the camera
dots,
down a little bit?
yeah,
Yeah.
some of these black
Okay.
dots are actually also green. Maybe if I remove this, you can see it. You see this green little dots over here, right? So these are cells that
Mhm.
has HIV integrated inside of the cell, and when they reactivate, when they start producing virus, they will turn green. So
Okay.
with our assay, we can check over time, and I hope you can, appreciate more or less
And an
that
assay
eventually,
is an
yes,
experiment of
it's an
some kind?
experiment. It's just
Okay.
basically to show whether our gene therapy approach is working. So here we have our treatment, and on the right side we have our control. So how it should be looking if you don't have any treatment. And as you can
Oh,
nicely appreciate over time, you get more and more of these green dots, meaning that there are more and more cells that are expressing the HIV proteins compared to our treatment in which we don't have as many, meaning that whatever we are treating in this
mm,
specific scenario, we do not get as much reactivation. So we are inside of the block and log approach
okay.
of hope.
So on the
Can you?
left, the green
Yeah.
is cells
Yes.
that
So
are
cells, exactly.
that have HIV inside,
Yes.
that are actively multiplying, producing more HIV.
So they are basically reactivating because if you know, or as all of you know, HIV is silenced, but for whatever reason, because of the different stimuli, they can reactivate and produce
Okay.
virus again, right? So when they reactivate, they start producing parts of the virus. Then, and one of these parts is actually these green fluorescent proteins that we can actually
Mm.
detect, and we see these green dots.
Can you explain, Fran, for
Yeah,
folks who are not familiar
yeah,
with how we use fluorescence in the cells with the virus, how that works, why it's green? Because we don't
right,
in the person HIV is not
right. So
green, right?
exactly. So in the person, the cells don't glow green, but we use a protein that comes from a jellyfish. Then we call it GFP, green fluorescent protein, and we basically modify the virus to have this protein inside of the
Whoa.
gene.
Okay.
So basically, we use it as a way to report activity.
Okay.
But HIV does not have GFP or this
So
protein. It's
to repeat that, you took a protein from a jellyfish that
but we
naturally
know
has
somebody.
this
Yes.
fluorescence,
Hmm.
and then added it to the HIV
Hmm.
so that it literally glows like a jellyfish.
Yeah, that's right. Pretty much.
No, all in a day's work. It's fine.
Just a regular Monday.
It's what some of the tools and the instruments that scientists have to utilize so that you can go on into the understanding the mechanics, right? So we modify by using these colors so we can see it, if you wish. It's kind of
So so the T cells that Julia was showing us and taking care of, you're using those, adding the HIV to the T cells, and then you're getting this.
So the HIV is already inside the cells.
In all of them.
Yes, in all of them. What
Oh,
we
okay.
want to see is whether the treatment that we are using
Yeah.
blocks the virus so that cannot wake up again.
So
So if you go back to the screening, whatever treatment that we are doing here is actually working because the virus cannot wake up.
are you
While
allowed
here
to
is waking up
speak
normally.
to what you've added on the right here to make the HIV less
So
expressive?
it's part of my gene therapy approach that we are developing. It's based on epigenome editor, which is basically kind of like switch on and off methodology. So you introduce kind of modifications that are not dangerous inside the DNA, and that tells the DNA I should not wake up or I should wake up. I should make proteins or I shouldn't. So we are kind of, kindly asking HIV to remain sleepy and don't wake up.
Yeah. So
Fascinating.
the T cell that Julia showed us, there's different types of T cells. They can be from donors and they have no HIV in them. But also in the line, we have what we call cell lines, right? And I guess you're working with a cell line.
Correct.
And in a cell line, there's an HIV that is what we call integrated. He is just hidden into the host genome, if you wish. And that's what the latent cell lines, and he's working with that to see is it reactivating, what we call waking up or not waking.
So, and this ties into Cure because with the method that you're using, you're hoping to make all the HIV essentially go to sleep, right?
Correct.
So that there's no green glowing jellyfish protein.
Exactly. So no green, or in terms of people living with HIV, that the virus cannot produce, cannot wake up, and cannot make you sick potentially.
So in this strategy for Cure, is it similar to I don't know, I want to say like it's not the best example, but something like chickenpox where you have the virus, but it's not expressing itself and it's not infectious.
I think the best example would be what we call endogenous retroviruses. So endogenous retroviruses are viruses that through the evolution of the humans we have got inside of our genome, but they do not cause any harm. They are just inside of us. Some of them are beneficial, some of them don't do anything, but they just stay dormant. So what we want to do is turn HIV into one of these viruses in the way that they are just dormant, they don't do anything to us, they're just there.
Yeah. And this assay or experiment here is kind of showing how you can achieve that.
Correct. It's a long way to go, but we are in the right direction.
Well, thank you for all the work you're doing, all of you.
Yeah, these three young people, I mean, kudos, hey, all their working very hard, very, very hard. So are there any questions?
Please, if you have questions, don't hesitate to drop them in the chat. I'm watching the chat. I know there's some questions about Cure timeline and stuff like that, which I'm going to save that for later in the conversation. But if you have any questions about what we saw here with the T cells and with the fluorescence, please let us know.
We are right here.
I know you have 3 Cure scientists here at the ready for you.
At the ready, yes.
Thank you, TV, so for the praying hands. I saw some people in the chat saying amazing. Mo Coelho said amazing. I think this is just really special for a lot of people. Fran Felix said crazy man to Fran.
Yeah.
Razors Club says great work to all involved. Jose says just want to say thank you for your work. Alexander Karimi says thank you for the presentation.
Yeah, we're really, really happy to, to connect with folks, and we want to do more of this. We want to really tell us, know what you would like to know. We can tell about ourselves. It's really important to address the science, but also all the human aspects of of living with HIV. And I know you're doing a fantastic job advocating for people. Yes, you have a question.
Oh, and I just want to add, for those of you who don't know, we have a very special guest coming on too, as well, the head of the lab, and she's going to be speaking as well. She's here now. So keep the questions coming.
Yeah, she's in Japan, actually.
Let me see if I can bring her on while everybody's here. Okay, Melanie, I accepted your request, so let's see. There we go.
There she is. Hey
Hey.
guys, so proud of you. You did so well. I don't have to say anything anymore.
Yes, they're really honed their skills in talking in an accessible way to community.
My connection might be not too strong, but I can see you all. But sometimes I think it freezes. I'm sitting in the middle of Japan here, and so amazed that I can join by Instagram. Wonders of social media.
So, can you tell us a little bit about who you are?
Absolutely, yes. So I'm the fortunate person who is able to work with all these amazing scientists every day. So I'm in Melanie Ott. I'm a trained neurologist. Started out in working in clinical medicine with people with HIV in the early 90s, and was so captivated by the virus that I switched my career, went to the States, did a PhD, and became a virologist and study HIV now. We're very invested in cure research. We have been working on HIV transcription for a long time, more than 30 years. And I'm really excited about the current time because I think we are really at a lot of new things are happening, and there's really a real progress might be in sight.
Fascinating. Thank you so much. I do have a question before we lose the other researchers, asking about how do you stay encouraged when experiments fail? That's a good question.
Man,
We try to avoid these situations, but I mean, what really helps is having a team, right? Like having good people around you that you can actually talk to, that actually also then remind you that, okay, maybe this one day or this one week you kind of screwed up, you, I don't know, wasted a sample that was really precious, something like that. But in the end, what really matters is the long term and the goal in the end of the road, right? So I think like having this supportive network around you really helps in that disappointing moment.
I mean, to add to that, having a life outside the lab helps. So when things don't go great, you can lean on that support system and lean into your hobbies and interests and just take some time to recover because it does suck. But you just brush it off and come back in.
Maybe I can say a few words. I think the point is really that science moves very slowly because there is a lot of failure. We do a lot of things to exclude things. But when you hit the jackpot and the thing is working and you have really nailed it down, I think it's priceless. So I think in some way it also compensates a little bit for all the failures that are going through. And I totally echo that working with good friends and good colleagues in a team is absolutely essential, helping to troubleshoot, but also think about alternative hypotheses and have a very open mind to really sort of interpret where the science leads you and not where you want the science to lead you.
Very good. And I'm sure it helps to have a great focused leader as well in yourself, Melanie.
Well, I try my best to cheer everybody up and help in the difficult moments. But I think we also have many very good moments. Especially discussing what the what the alternatives are, what the interpretation of the results are, and I think this is incredibly enriching. Because I think at this level we are all colleagues. I think we're doing different parts of the job, in terms of doing the work in the lab, or me finding the funding and and helping steer everybody in the right direction, or in the same direction, let's say. But I think the key is that it's an incredibly interactive and collegial environment that can only succeed if you really have diverse opinions, diverse minds, and diverse people coming together and working on one single problem.
Speaking of funding, one of our viewers is asking, how are you funded?
Who's taking that one? Melanie?
Very much
Okay.
funded by the NIH. I think there's different ways to fund. I think Gladstone is a non-for-profit research organization. I think we are 90% funded by the NIH. We also receive donations and have dedicated philanthropists who help us in our work. And then each of these talented researchers also apply for their own funding in terms of fellowships, in terms of grants, in terms of different opportunities that are tailored. So it's a constant looking for funding, but it is also and a constant being vetted by funders that helps us stay the course and move forward.
And going back to your comments about failure, Mr. Mercer Hall says being able to fail fast, fail forward, and celebrating the small wins are all essential.
Absolutely.
And I want to add too, your note about the exclusion that failure brings. It's like, to kind of break it down, if you have numbers 1 through 10 and you're trying to figure out which number is the number that you want, maybe you start out with all the even numbers and you realize that failed. And now you know it's one of the odd numbers. Well, is that really a failure? Because now you've just excluded a certain number of numbers and you're narrowing it down the more you go.
Totally. We exclude hypotheses most of the time, or we confirm them. That is the one thing. There's also technical failure that we have to take into consideration. These are highly complicated and sophisticated assays that are being set up in the lab, and they need to be vetted, they need to be established, they need to be controlled, they need to be ironclad in order to be trustworthy. And I think so there's different levels, but you're totally right. I think sometimes we start off with a hypothesis, and that's very important that you have a hypothesis, because otherwise you go into many different directions. And my people in the lab know that I'm very strict on having a model where you to go into an experiment, so that you can interpret the data correctly and can modify your model or your hypothesis and move forward and test and exclude the as you said, the even or the odd numbers, so that you narrow it down to the right ones.
Bezad Haider Shemas asks how to donate, please.
There's a donation button on the Gladstone website. We can send it to you. You can
Maybe
also
Ed, can you put it in the chat?
Somebody has to put it in the chat, because I cannot handle Instagram and
No.
anything else. I'm already very challenged with my Instagram. I was coached by my 19-year-old daughter. She was very helpful, and I'm really excited that it works so well.
And
Thank
that's the
you.
thing too, is we need more involvement from science in social media to reach the younger people.
Absolutely. I think it's a very important time, given that a lot of false information and is circulating unrestricted on social media. And I think we need to make sure that we also circulate what we think is the correct news, and get it to the right people. I think it's a very, very important time. It's a very important task, and having you all online here is incredibly important for us. Thank you.
Oh, thanks for everyone. I have multiple people putting the links in the chat if you want to check it out. Yeah. Yeah, I'm always telling people, if qualified people, real experts, aren't in this space having the conversations, talking about these things, then the unqualified pseudo experts will. Someone's going to fill this space and someone's going to talk about it. So if the qualified people aren't doing it, then the unqualified people will, and that's what has happened so often.
Yes, and that's why we're very grateful for you, Raif, because to give us, a platform to make sure that we're also being heard.
Of course. Okay, so I do have some questions from
Can I say
earlier.
one more thing?
Yeah,
I think the other
please.
thing that's so important about these live, that you all learn that we are all normal people. Scientists are not these like zombies or something. Scientists are very real people. They have lives outside the lab. They get frustrated. They need support, emotional and otherwise. And I think this is very important, because we're not living in an ivory tower. We are, connected with everybody, and we do, the work as well as we can with the best intention, and with a very strict training and analyzing the data the way they are, and not sort of trying to put our hopes and wishes into them.
Yeah, yeah.
Great. Thank you for that.
Words.
Getting snaps from the
Snap,
lab.
snap, snap.
Okay, so one of the questions I had earlier is how do you handle the fast virus mutations of HIV with treatment? How do you like stay up with that?
It's a very, very good question. I think we are very aware of the virus is very versatile in the way that it can immediately, it constantly produces mutations that can then help evade treatment, or, the efforts to, produce a vaccine. Here we have actually the biggest problem with all the different variants and the different quasi-species of HIV that arise. I think there's many different ways of tackling this. I think when you come to the therapeutics, we know that we have to combine different therapeutics in order to make it harder for the virus to, move into many directions at the same time, and in order to inactivate the treatment or build resistance against it. So I think drug combinations. I think in the vaccine field, a huge amount of effort is being focused on conserved areas of the virus that cannot mutate so fast. And we specifically are focused on the transcriptional machinery that produces the viral RNA. And that is something that the virus cannot afford to mutate so much. So I think these are sort of conserved areas versus very highly variable areas. For example, the glycoproteins on the surface of the virus, they undergo extensive mutagenesis, but other areas in the genome are less prone to accepting or tolerating any mutations. And so I think another way is to focus on these conserved areas. And again, I think a treatment is never going to be one pill or one single medication. It's always going to be a combination. And in the lab, we also not only working on drugs per se, chemical drugs, we also work on gene therapy approaches, on approaches that are complementary to these other approaches, in order to be really reaching every viral isolate that is out there.
And Neo the Leo in the chat says, I feel like a lot of HIV researchers retired in the last few years. Is that true? Should we get more young people involved in the research?
Right
Always.
here.
Yeah, always.
I
Absolutely,
would
you're
love to.
looking at the future of HIV research, and I think the future is bright, as long as the support is there.
Yeah.
Yeah, but it's totally true. I think there was a wave of researchers that got into the field, 40 years ago, as young people, and they're, sort of cycling out now, and I think new people are coming in, and we're very proud of our trainees and our young people.
And so, speaking about how quickly HIV can mutate, I get a lot of questions about like why is it taking so long to find a cure? Here we are, kind of curing these other diseases and things, and yet this one, you know, there seems to be no progress. Is that related to the mutations? Why do you think that is?
Well, it's a very complicated first of all, I think we should acknowledge that actually antiretroviral therapy was an amazing breakthrough, and I think it has helped enormously. It's not a cure, but it was, an amazing progress. So that is something that needs to be acknowledged, because I have still, lived during the times when we had no antiretroviral therapy, and it was a very, very, very depressive time. And so now the point is that antiretroviral therapy takes care of 99% of the viruses in the body, but it cannot touch these 1% that is basically the long-lived reservoir that is remaining. And these cells seem to be very heterogeneous. They seem to be in different, compartments of the body, and they're very hard to study because it's only this 1% that is remaining. And so we have spent a lot of time in trying to mimic it in the lab, sort of the conditions, so that we can test and develop tests to do this. We're also spending a lot of time to isolating these cells, from people, in order to study them. And the difficulty here is that we have a lot of it's really like a needle in the haystack. It's a very small percentage, because we have a lot of cells that carry viruses, but these viruses are not replication competent. They're called defective, so they're on their way out. But I think these are not what we want to study. We want to really go for the ones that are replication competent and can cause harm when we take antiretroviral therapy away. So I think there's multiple issues here that need to be addressed. And then, there is a lot of efforts that have been going into many different ways how to address these remaining cells. Some think we have to reawaken them in order to eliminate them. Others, like us, think we should not reawaken them. We should actually silence them down and make them like part of our genome, like endogenous retroviruses. And others think that we need to develop immunotherapies against it. And the truth will be probably in a combination of many of these different approaches. And that's where we are currently. We have put forward a lot of different approaches. We're very excited about ours. Others are very excited about theirs. And now we're testing them in animal models and in clinical studies, which takes always a lot of time. And I think eventually we will have to potentially combine them in order to evade, mutation of the virus, but also evade and really reach everybody, but also potentially personalize it to whatever is happening in individuals. So that's why it takes a little longer.
I have a comment from North African 747. They said, I don't know you all apart something, but I love you all so much. I think they're saying from someone who is HIV positive, has been diagnosed 1 year ago, and put 3 hearts.
Was sorry about it, and 3 hearts back.
Yeah.
McSilva 2446 says, I'm a family doctor in Brazil, and we know how hard it is to show a person who gets infected that life continues normally.
Very true.
And earlier, just so you're aware, Melanie, someone said, Jermaine Brown says, my cousin is a nursing student and would like to be a part of the research. Can anyone help me in getting her on the team? And so I think Eb sent a link.
Absolutely. We offer internships, and we have, many of opportunities, shadowing opportunities, other things that interested people can explore.
And that's why it's so important that we have this young team here that's, being visible for everybody to show, like, young people. It's the young people who need to come and join and, add.
Totally, totally. I think the work is not over. We are still very much in the middle of it, and I think we're preparing for the last push.
Hmm. Spartan Lad says, diagnosed in 1985. Thank you for working so hard to find a cure and treatment.
Thank you.
And my personal question, I guess, is for the cure, do you think it will be you'll discover something that will cure people right away? Or do you think it'll be maybe like a progressive thing, kind of how we've seen HIV treatment grow and progress, where it was multiple pills, maybe a couple times a day, to once a day, to one pill once a day, to now long-acting injectables? How do you think that cure might come about?
Well, if I may answer, I think it's not going to be immediate, and it's not going to be one, silver bullet here. I think, as you see, we are inching forward on many, many different fronts. I think already introducing art as early as possible is an enormous benefit and has helped. And I think now we are working on how to best complement art with other treatment options in order to make sure that we can take people off antiretroviral therapies for longer and longer times. I think that is really the goal. It will not happen for everybody at the same time, but already now we have some clinical trials and some treatments where individuals in groups do not rebound after, being taken off of antiretroviral therapy. So there is, sort of small successes, but they are not yet, really felt universally with everybody. And so we will inch forward, as you said, with like we did with antiretroviral therapy, to optimize these treatments, to put them into standard treatment approaches, and to especially test how different individuals react to it. And I think that will take time, but I would say we are making progress already, small progress, but I think this combined will help in the future.
Yeah. Julian Maurizio says, hi from Myanmar. Thanks for all of your efforts.
Wonderful.
Yeah, we have team members.
Kushea,
Oh,
go ahead.
No, I want, because I hear you're joining us from Myanmar, and this is the amazing power of the internet, right? We have people from all over the world, and our research team is also all over the world. We have colleagues, and the whole family is incredible. And we have amazing folks in Brazil, in Sao Paulo, in Uganda, in Germany, and it's really a privilege to be working with all these folks, and very connected to communities as well. The scientists in these research team are also very connected with their communities locally.
Have you guys talked about the community work a little bit? I think it's really fascinating.
No, we have not. No, I think we wanted to get this opportunity to ask as many scientific questions as possible. But depending on the questions in the chat, yes, I'd be happy to
I
talk
do
a
have
little
one.
bit.
I have one question from H61 Graham saying, can a cured, and of course this is all postulating, but can a cured person be reinfected? That's an
Well, that
interesting
is
question.
actually a very, very important question, and it's something that there's a lot of emphasis on this, that the cure strategies that being developed would prevent future infections. We don't think that the immune response is sufficient to prevent you, from being reinfected, but I think there's a lot of effort being focused on to develop strategies that would help with this, and so this is a very, very important question.
Yeah,
Hi, Jose in Mexico. Go ahead, Patricia.
No, I just wanted like to piggyback on what was just said, when you think about cure, and it's not just one isolated thing, it's like really a continuum of care. It really goes in hand with prevention, with vaccine, with treatment, you know, seeing the person as a holistic being. And once you're going to have a cure, you want to make sure people will not acquire the novo, the virus. And how do we work along with the new long-acting injectables, and all of the different research streams will interface. It's very important.
And it seems like with what you were saying, Melanie, if you're targeting the part of HIV that is constant, that's the same in all the different strains, and you're able to fight that off, then no matter what strain, because I get that a lot too. What if you get a different strain of HIV and you've been cured from one, but then can you be infected by another? But if you're kind of resistant to all the strains, then that solves that. I
Well,
have
I
a
think,
question. Oh,
yeah,
go ahead.
no, totally important, because I think a cure is going to be only successful if it targets all the strains, and I think that is what we are aiming at.
Yeah, snaps to that. TVSO Sam says, is there a virus from human or animal genome history that parallels HIV in a way that scientists or your research team are studying to glean potential cures?
I didn't get the beginning. Was it about animals?
Yeah, is there a virus from human or animal genome history that parallels HIV in a way that you're
Well, I
able to
think
study it?
very, very important question. I mean, we have, simian infectious, immunodeficiency viruses in monkeys that are often used as a model system. But we at Hope, we are very focused on a very unique fact in the human genome, which is that a big part of our genome, actually the bigger part of our genome than, for example, the part that encodes for our proteins that are, determining our day-to-day life. I think a big part is actually ancient retroviruses like HIV that have invaded our genome millions of years ago, and that have basically decayed in there and have become pseudo silent, or at least a part of us, that our body is dealing with very successfully. And so I think we are studying exactly these human endogenous retroviruses in our genomes to learn how we can accelerate the path of HIV to become exactly a fossil like this, that is basically no longer an immediate threat, and that we can actually very happily live with. And I think that is what we're studying very actively. We have experts on the team that are only coming from that direction, and we have experts in the team that are coming from the HIV direction, and we think that this is a very good combination to learn from each other, to really apply things from one to the other in order to really find something that is working for HIV, again, all HIV, to become harmless.
Great, thank you. Well, I want to take the last 3 minutes to just, first of all, say thank you for all of you for giving your time and your energy and giving us a sneak peek into a place that most people never even get to peer into, and certainly don't get to ask questions of. And I just want to kind of open it to you all to see if there's any last words or comments that you'd like to make.
Well, I'm very grateful for you all, scientists and out there in the community, and I wish you all the best. I talked a lot. I give the last words to my people.
It is an honor to do this work, and I'm really looking forward to seeing more young people coming into this space and to more opportunities to connect with the community and talk about cure research as it goes along.
So for me, it's also I'm very grateful to do what I do and to have the opportunity to connect with you all through the screen. I don't have any Instagram or anything like that, so actually it's a great opportunity. And also for me, at least, it makes me feel that what I do matters, and it makes me work harder and get stronger every day, and stand the frustration so we can make it work all together. I'm really looking forward for the future.
Yeah, I can only second that really. And it's actually for us as researchers, we spend like days in the lab in these kind of areas, and it's so good to see people out there that show interest in our work. And we also see that actually the work that we are doing is important for like people out there as well, and like having this connection through these events. We're really grateful to have this here. So thank you all for coming here and joining us.
Thank you.
Yeah, I can only shout out to you, Raif, for your dedicated advocacy for all you do on the social platforms. I'm not a social platform person, but I think it's an incredible way to connect with folks. And I want to shout out to communities, because HIV, yes, it's the virus, it's the lab, but it's also mental health. It's also access to healthy food. It's also having communities around you that support you and care for you and do not reject you because of the stigma. And I want to, do a big shout out to the San Francisco AIDS Foundation, who's our community partner on the HOPE project, because we have, I was talking about the HOPE family. We have really, it's really a blessing, right? It's the power of all of us working together, and everybody has skills and expertise. Yes, we have the lab expertise, the scientist expertise, but community members have their lived experiences, expertise, and all that you bring to us. We learn as much. I'm kind of in between the two worlds, but I hear it. I hear it from both sides. We're learning so much, and I'm so thankful for you for taking the time today from your experiments, and for all of you who've joined. Melanie, for you from coming from Japan, and for Sydney, who's been holding the phone. She's gonna have a really strong arm. Yeah. Sydney is
Yeah.
also, she does all the coordination for the team. So she's really part of all the aspects of our project.
And don't worry,
Very
folks, this isn't just a one-off. We're gonna keep following their work. We're gonna follow Melanie and all the researchers, and I've interviewed some of them as well recently at their poster sessions. So once we get that edited, that'll come up. This will be an ongoing thing, so you can stay part of the journey and get updates as we go along.
Yeah. Take good care, everybody.
All right, thanks everyone.
Thank you, Raif. Thank you,
Bye.
everybody. Bye bye. Happy World AIDS Day.